Well, good morning to everyone. It's now just after 8 o'clock in California time. I apologize, I was a little late this morning, but let's begin. This morning, I'm going to tell you about an effort that I've been involved in and which has engaged in some of my research activity concerning a serious neurodegenerative disease that has afflicted over 6 million people around the world, and it is Parkinson's disease. So let me first tell you about the nature of this disease as it affects so many people around the world. And then I'm going to delve into the basic science of the disease, what we know about some of the genetic forms of the disease. And then I'll tell you about an international consortium of investigators who have come together to study in a collaborative mode how we may tackle this terrible disease. So let me begin. Parkinson's is a pandemic. It affects now almost 8 million people around the world, and it's projected to expand to an even greater number as time goes on. Although about 30% of the patients afflicted with Parkinson have some genetic predisposition to the disease, it's still largely a sporadic disease that has no known cause, though it may be influenced by environmental toxins and by behavior and such as smoking or longevity, industrial byproducts. The disease knows no geographic boundaries. It is universal, much like the current pandemic. The rise of incidence is particularly acute in China, and it's predicted that in the next decade, over half of the new patients who suffer from Parkinson's will arise in China. The disease is complex in its origin and progression. It appears to have different paths of presentation and rates at which it progresses. Some people progress very rapidly, have dementia at an early age, and suffer a very low survival over a short period of time. Others can live for a much longer period of time with a very slow progression of disability and little dementia. Nonetheless, overall, almost 30% of patients who suffer from Parkinson's have some dementia, which seriously complicates their life in addition to the movement aspects of the disease. So this may be a reason why it's often considered a spectrum disorder. Many different forms of the disease appear to converge on a pathology that's associated with a small region of the brain called the substantia nigra. This is the place in the midbrain where dopamine is produced and secreted by dopaminergic neurons. And let's consider some aspects of those neurons now. Here is several images. First, of a section of a brain taken from a normal person who died for other reasons. The cells that make dopamine also produce as a product a colored substance called melanin, which gives these cells a dark appearance that can be readily seen in cross sections of the brain of someone who died. Parkinson patients, by the time they present with the movement disorder symptoms, have often lost over half of the dopaminergic neurons. And by the time they die, what is often seen in sections of patients who have succumbed is the absence or greatly diminished appearance of melanin in this region of the brain, the substantia nigra. Now, about 100 years ago, a British clinician noticed in sections of the brain where patients had died, particularly in the region of this substantia nigra, there appeared within these cells inclusion, dark stain inclusion bodies that we now know to house protein aggregates, in particular a protein called alpha-synuclein. the crude representation of the linear sequence of the protein alpha-synuclein is shown on the bottom. It's a small protein. It's a relatively unstructured protein. And there are genetic forms of the disease that result from mutations in the amino terminal portion of the protein shown here. These three mutations turn up in patients. These mutations tend to promote the aggregation of alpha-synuclein, and it's believed that that aggregation may initiate the formation of these structures that have been called Lewy bodies in honor of the British clinician who first discovered them in these sections. Now, it's possible in more recent research to reproduce in neuronal cells grown in the laboratory. It's possible to reproduce these structures in a cultured cell when these cells accumulate an aggregated form of alpha-synuclein. And you can see an image, a fluorescence image of such a cell shown in the upper left-hand corner, where the synuclein aggregates are decorated with an antibody that has a fluorescent tag that allows one to visualize these structures. In a thin section electron micrograph of such cells, highlighted in false color, you can see the material that tends to accumulate in these structures. It consists of fiber-forming proteins, of membranes such as the mitochondrion and other organelles, as well as protein aggregates, including but not limited to alpha-synuclein. Now, the question that we have yet to address is whether these aggregates are the foundation of the pathology or merely a byproduct of the pathology. That is to say, is the accumulation of such structures in neurons, particularly dopaminergic neurons, does that result in their death, or is this an attempt by the cell to contain the protein aggregates and to reduce the pathology? Nonetheless, the cell is inevitably propelled to a premature death. Now, some years ago, a German clinician observed in sections of brains taken from patients who died very early or in middle ages or in old age of Parkinson's disease. Dr. Brock observed an apparent progression of these structures, these Lewy bodies, initially from the stem of the brain, possibly having moved from the gut through the vagus nerve into the brainstem, and then subsequently migrating to other regions of the brain that affect motor control. And finally, for a subset of patients suffering from cognitive decline, Dr. Brock observed an apparent spread of the Lewy bodies to cortical regions that affect more than just movement, but also control cognition. So it is believed, though it has not yet been proven, that the alpha-synuclein can spread within the brain. And the question has been, how can it spread within the brain when the protein itself is a cytoplasmic protein, lacking the kind of a signal peptide that would allow it to be secreted outside of the cell? Now, many investigators, including several in my lab, have found that cell cultures that express alpha-synuclein and overexpress a chaperone protein that appears to promote the secretion of alpha-synuclein can be reproduced in the laboratory, where we can now study what we call unconventional secretion of alpha-synuclein. And let me now show you some results from my laboratory, and which results that have been confirmed in other laboratories that provide a possible route for the unconventional removal of alpha-synuclein from the cytoplasm to the outside of the cell, where that may then spread to neighboring cells, be taken up, and promote a progression of aggregation and unconventional secretion. To summarize what I'm going to show you in the next few slides, we and others have evidence that alpha-synuclein produced in a normal cell engages an oligomeric form of a protein called DNA JC5. I'll tell you more about this protein in a moment. A protein called DNA JC5 that is itself attached to the surface of a membrane called the endosome by a lipid group, a palmitic acid lipid group that is covalently attached to the DNA JC5 protein. And we believe, but have not yet demonstrated, that this oligomer may present a channel through which alpha-synuclein may pass from the cytoplasm into the interior of this endosomal membrane. Let me show you some of the experiments of a postdoctoral fellow in my lab, Dr. Shenji Wu, who has investigated the unconventional secretion of alpha-synuclein when it is expressed in a normal conventional cell line that we use and many use in the laboratory called HEC293 cells. Here now is a depiction of the a crude representation of this protein DNA JC5. The N-terminal region has homology to a family of proteins that is found in all organisms from bacteria to man called DNA-J. DNA-J is a chaperone that facilitates the nucleotide hydrolysis by ATP, a variety of ATP binding proteins. And that domain is shown here at the N-terminus of the DNA JC5. There is, in addition, a relatively unstructured C-terminal domain. And then in the middle of the protein, there's a very important region with many cysteine residues. It's called a cysteine string domain. And these cysteine residues are the sites for covalent attachment of palmitic acid in a thioester linkage connecting the fatty acid to the protein, making it somewhat hydrophobic. A human disease that affects this protein is represented by a mutation at this position. And this mutation is enough to prevent the palmitylation of this DNA JC5 protein. Now, here we see an SDS gel of the mature form of DNA JC5 with its palmitic acid group attached, the protein of slightly lower mobility on an SDS gel. And then we also see a version of the protein that has no palmitic acid and allows the protein to migrate more rapidly on an SDS gel. Now, in one experiment that Dr. Wu has conducted, we have taken the cell line that I mentioned, HEC293 cells, and have transiently transfected into the cells a fusion protein where alpha-synuclein is linked via its C-terminus to a protein called N-luciferase, an enzyme that can be sensitively detected because in the presence of a substrate, it generates photons that can be measured quantitatively. So it's a very convenient way to monitor the production and secretion of alpha-synuclein because the fusion protein is secreted as efficiently as the normal protein. Now, when these cells are transiently transfected by introducing a gene, a copy of the DNA JC5 protein versus a control where only the GFP gene is introduced, we see these cells stimulate the secretion of alpha-synuclein fused to luciferase about threefold after as little as 16 hours after introduction of the DNA JC5 protein. By a day and a half after this transfection, the level of N-luciferase activity representing alpha-synuclein rises very substantially in the growth medium outside of the cells. So a huge increase in the unconventional secretion of this protein promoted by the expression of this DNA JC5 protein. Now, we can further demonstrate this by directly evaluating the DNA JC5 protein in the medium or in cells after this 16 hours or 36 hours. And so if you will focus first on these lanes in the upper left, these are cells that are all expressing just normal alpha-synuclein without or with the expression of the DNA JC5. The lanes on the far left represent the conditioned medium that is harvested after the cells are removed, or the cells themselves harvested for evaluation by an SDS gel immunoblot. You see some stimulation of alpha-synuclein evident after 16 hours. You see quite a bit of the palmitoylated DNA JC5 protein itself now secreted into the growth medium, all exclusively in the palmitoylated form, whereas within the cells, both palmitoylated and non-palmitoylated protein are detected. Now, more importantly and more impressively, after 36 hours of transfection by the plasmid containing DNA JC5, there is a substantial increase in the appearance of alpha-synuclein by immunoblot detected in the medium. Now even the non-palmitoylated as well as palmitoylated DNA JC5 is detected in the medium. So a substantial increase both of the fusion protein to synuclein and the wild type normal protein can be seen by overexpressing DNA JC5. Now, looking more closely at cells that are labeled so as to visualize where the alpha-synuclein and where the DNA JC5 reside, Dr. Wu constructed a cell line that has a mutant protein called RAB5 with this mutation. The virtue of this mutation is that cells accumulate giant endosomes that are much more readily seen in the fluorescence microscope images than is evident in cells that do not possess this particular mutation. So in cells that have this mutation, but which are not overexpressing DNA JC5, you see some DNA JC5 contained within the interior of these endosomes. Some have quite a bit, some have a little, but the DNA JC5 is less pronounced in these cells. Now, if you look at the same cells that are expressing a fluorescently tagged form of alpha-synuclein, but without the overexpression of DNA JC5, you see the synuclein protein distributed throughout the cytoplasm. And specifically, you can see these endosomes here marked by another fluorescent dye attached to this mutant protein. You can see these endosomes, but they lack any alpha-synuclein that has managed to get into the inside of the organelle. When DNA JC5 is overexpressed, this protein can be detected inside the endosome. But when it's not overexpressed, you don't see any alpha-synuclein. Now, the next experiment shows when both the RAB5 tagged protein is used to visualize endosomes and DNA JC5 is overexpressed, as well as alpha-synuclein with a fluorescent tag. And you can see now examples of not only DNA JC5 internal to the endosome, but you can see examples of alpha-synuclein internal to the endosome. So this is what we believe is the precursor of the secretion of alpha-synuclein. It enters from the cytoplasm into the endosome, and if the endosome then travels to the cell surface and fuses with the membrane of the surface, this alpha-synuclein and the DNA JC5 may be secreted. it's also possible, as you see on the right, to demonstrate that in lysates, gentle lysates of these cells, some fraction of the alpha-synuclein is resistant to treatment with a protease that would degrade the alpha-synuclein if it were on the outside or on the surface, exposed on the surface of endosomes. But you see some fraction of the alpha-synuclein is resistant to that protease treatment unless the membranes are dissolved with a detergent, which allows all of the protein to be degraded by the exogenous protease activity. So now I'm going to return to other studies on the nature of the cells that suffer from different forms of the genetic disease, Parkinson's disease. And one of the major sites where mutations have a very serious effect is on the removal of damaged mitochondria that may develop as the dopaminergic neuron does its business. The dopaminergic neuron is highly active. It is engaged in interactions with each dopaminergic neuron may interact with 1 million different nerve cells in the brain. And so it is highly dependent on the mitochondrion, the powerhouse of the cell, to manufacture ATP to allow the cell to do its business. Now, mitochondria are beautifully designed for this purpose, but they must remain intact because damaged mitochondria can cause a cell to die. When a mitochondrion is damaged by its high use and turnover, the DNA that is contained inside of the mitochondrion sometimes is released into the cytoplasm of the cell, and that may cause the cell to react with a pathway that ultimately leads to the death of the cell. Likewise, in the production of ATP, the mitochondrion generates oxygen-free radicals that are held inside the mitochondrion, eventually to be reduced to water at the end of the process that makes ATP. And if the mitochondrion is damaged, these oxygen-free radicals can escape from the broken mitochondria, and they can enter into the cytoplasm or the nucleus of such a cell and do a great deal of damage, including breaking chromosomes. So the mitochondrion is a terribly important organelle, but it's equally important that its damage be contained. And there is a process that all cells that have a nucleus have evolved to make sure that damaged mitochondria are disposed of or in an orderly process. When damaged mitochondria accumulate in the cell recognizes the damage by attaching a special tag, a small peptide that I'll tell you more about in a moment, a small peptide tag on the outside of the mitochondrion that causes that organelle and other organelles to be swallowed up into an organelle called the lysosome, which then degrades these damaged membranes and all of their contents by attack by enzymes such as proteases and lipases and nucleases. And these lysosomes then can recycle the amino acids and sugars and carbons that are generated by that degradation back into the cytoplasm for normal metabolic use. So this is a process called autophagy, or as it pertains specifically to mitochondria, it's called mitophagy. Mitophagy is an essential process for cellular homeostasis. Now, it turns out that in Parkinson's disease, there are 2 genetic forms of the disease that result in damaged mitochondria not being tagged, which then precludes their capture and degradation within the lysosome. These 2 genes are called PINK1 and Parkin. and I'm going to tell you just a little bit about both of them. PINK1 and Parkin cooperate to tag the surface of damaged mitochondria with a special little peptide called ubiquitin. Ubiquitin is a small peptide. It's basically a signal that the cell uses to mark those structures, organelles or protein molecules, that the cell determines must be degraded. And so damaged mitochondria accumulate these kinds of little tags, and that allows them to be enveloped by a membrane called the autophagosome. This autophagosome completely encloses the damaged mitochondria, and then the autophagosome with its cargo joins by fusion with the lysosome, resulting in the attack on all of these membranes by enzymes in the lysosome that degrades these structures to amino acid sugars and carbon units. So if either if you have a family history of Parkinson's, some families have a mutation in either the PINK1 gene or the Parkin gene, and those mutations lead to a failure of decorating the mitochondria. The damaged mitochondria persist. They may leak their DNA or oxygen-free radicals, and that causes the cell to go into a pathway of cell death that commits the cell to dying. And this may be the explanation for those patients who have mutations in PINK and Parkin. That may be the explanation why the most vulnerable cells, the dopaminergic neurons, die prematurely. So here is now a visual demonstration of what happens when a cell is exposed to a chemical that damages the mitochondrion. On the top panels, we see regular tissue culture cells that are not treated. They can be labeled with Parkin, but it's not particularly evident in these cells, except diffusely in the cytoplasm, or they can be labeled with a tag, with an antibody, with a fluorescent marker that measures the presence of this ubiquitin tag. And you don't see any labeling of mitochondria under normal circumstances. However, if you damage the mitochondria in these cells with a chemical that induces a change in the membrane potential upon which the mitochondrion is dependent for survival, within a very short period of time, Parkin begins to accumulate on the surface of mitochondria, which you can see here as these aggregate forms are shown highlighted in the cell. And these coincide in position to ubiquitin, which can also label the same structures, and the two now overlap quite extensively, showing that ubiquitin tags are bound on mitochondria that have the Parkin protein. Here's a little bit more detail about what goes on when these cells are exposed or not exposed to a chemical that damages the mitochondria. In the healthy state, this protein called PINK1 is made in the cytoplasm. It partially enters the envelope that defines the boundary between the mitochondria and the cytoplasm of the cell. And an N-terminal, amino-terminal portion of the protein is clipped by a protease inside of the mitochondrion. And after this clip is made, the protein then simply pops out. It withdraws from the membrane and is subject to degradation by an enzyme in the cytoplasm of the cell that chews this up into amino acids. So the protein enters, but quickly leaves and is not lodged on the mitochondrion. Now, in contrast, if the mitochondrion is damaged, then this process of entry is halted because this amino-terminal portion of the protein fails to become exposed to the protease that would make this clip. And as a result, this molecule remains lodged on the surface of the mitochondria, and it stays there where it interacts with parkin, which is a form of ubiquitin that becomes covalently attached to the pink 1 protein and persists in that location for a very long period of time until the mitochondrion can be swallowed up and degraded. Now, one final genetic form of Parkinson's disease that is also apparently leads to the premature death of dopaminergic neurons is highlighted in a broad form here. The gene is called LRRK2, LRRK2, and the gene encodes an enzyme called a protein kinase. This kinase acts to put a phosphate group on different target proteins. The LRRK2 protein is very poor in its kinase activity. It has a very low activity, but mutations in patients, some patients who suffer from Parkinson's that happen to be in this gene, turn the kinase from a poor enzyme into a better enzyme. So the mutation actually turns on the kinase in an uncontrolled manner. And as a result, this kinase becomes more active than normal, and it transfers phosphates to proteins that it shouldn't be touching. So it's acting inappropriately. Now, a team in Scotland and in California has made the following proposal about how this inappropriate kinase activity of LRRK2 may lead to the death of dopaminergic neurons. It turns out that the highly active form of LRRK2 transfers phosphate to a subset of proteins called Rab proteins. These are small GTP binding proteins that control avenues of membrane traffic in cells. And when these Rab proteins have a phosphate on them, they, in one case, act to block the production of a structure on the surface of some cells called a primary or sensory cilium. And in the absence of that cilium, the cells that make dopamine fail to properly communicate with other nerve cells in a region of the brain called the striatum. And that failure of communication leads to a failure of the nerve cells in the striatum to produce a growth factor called GDNF that is required for the ongoing health of the dopamine neurons. So let me highlight this again. In the presence of the LRRK2 mutation, this primary cilium on a cholinergic neuron in the striatum fails to make this primary cilium, and that failure blocks a signaling pathway between the dopaminergic neuron and the striatal neuron, which is called the Hedgehog Pathway. I won't bore you with the details of that, but suffice it to say, in the absence of that communication, the cholinergic neuron doesn't see the need anymore to produce this growth factor. And in the absence of that growth factor, these dopamine neurons cannot survive and they die progressively. So a simple mutation interrupts a communication pathway that would be essential for the survival of the dopamine neurons. And as a result, they die more rapidly and patients succumb to Parkinson's disease. Now, let me, in the last few minutes, tell you about an effort that I have become involved in to try to align many of the groups around the world who are engaged in a basic science approach to Parkinson's disease. I became interested in this area for two reasons. One is part of my own work concerning the unconventional secretion of alpha-synuclein may relate to the
spread of Lewy bodies in the brains of patients who suffer from this disease. But I also had a personal interest in this because my wife suffered for 20 years with Parkinson's disease, and she died about 5 years ago. After my wife died, I was approached by a representative of a major philanthropist to consider initiating a collaborative research program to engage many more scientists around the world in an effort to collaborate and to communicate across these boundaries about their new results to try to reinforce progress in this very difficult subject area with so many different genetic components to it. The aim of our program was to build a collaborative research network. Let me tell you how this has developed. Just to emphasize the difficulty of the problem, there are many different reasons that ultimately lead to the death of a dopaminergic neuron. There is a failure of this protein alpha-synuclein, which may misfold. There is a failure in the systems that degrade normal proteins inside the cell, or there is a failure of mitochondria to be degraded. There are also inflammatory processes that rely on a form of immune system within the brain that may initiate the disease or certainly exaggerate the disease as it progresses. And then there is ultimately a pathway of cell death called apoptosis that leads to the premature demise of the dopaminergic neuron. We can view each of the influences as a piece of a puzzle, although the puzzle is much more complex than this. It's a puzzle that may have thousands of pieces, and finding a way to put such a complex puzzle together, we believe requires the integrated efforts of many scientists, not just those working in their own laboratories. So here's what we've constructed. We've constructed an effort that is based on 4 primary goals. The first goal is to understand all of the genes and the proteins that they encode that have been implicated in genetic forms of the disease. I mentioned that there are many genes, now over 20 different genes in families with the disease, and how these genes and their proteins interact with one another, which pathways they inform that lead or that converge on the dopaminergic neuron remains an active area of investigation. Another is this inflammatory process that may initiate or exacerbate the demise of dopaminergic neurons. We think that there are there's an immune system in the brain, and the body's own immune system may inform this process. We also need to know more about the circuits to which dopaminergic neurons engage. I mentioned to you that a dopaminergic neuron is enormously complex, perhaps the most complex neuron in the brain, with a single cell body aligned to a nerve terminal that has 1 million different endings that connect on different contexts. That cell is poised to do a great deal of work to organize communication in the brain. And as a result, it is particularly vulnerable, and it may die because of influences from many different sources. So the circuitry would be important. Another and final puzzle is the often very long prodromal period of time that precedes the onset of the disease. We know that patients who eventually present with the movement disorder often experience a poorly developed sense of smell, or they have problems in digestion in their gut. Of course, these are not diagnostic of Parkinson's, but very often patients who present with Parkinson's report that they have had such ailments in the past. One syndrome that is particularly diagnostic of progression to Parkinson's is a form of disease called REM sleep disorder. And patients with REM sleep disorder fail to control their dreams and nightmares. And often, as they're sleeping, they live out nightmares, and they can become violent even while asleep. Patients have reported
finding themselves thrown out of their bed because of their violent actions, or they can harm themselves or their partner by flailing around because of a failure of somehow controlling the acting out of nightmares. And it is amazing that something like 80% of patients who present with such symptoms progress within a decade to the movement disorder aspects of Parkinson's disease. So that is quite remarkable, but we know nothing about what may be happening in patients with this prodromal syndrome. Why is it that this progresses to Parkinson's disease? Well, let me tell you where we stand in my last slide. My second to last slide is we have progressed by identifying teams of investigators, not merely individual professors, but teams of investigators where these teams have already been engaged in collaborations. We want teams who know the give and take of a collaboration to extend their collaboration now to an entire network. So we looked when people were applying for those individuals who had already formed a team to work on Parkinson's or something else that may be relevant to the disease. Of course, we want to attract diverse talent. We didn't want to be limited to those already working on Parkinson's. We wanted also young investigators at the outset of their career who would bring in new ideas that would inform the more senior people of new directions that they may take. We have focused the effort on the 4 themes that I mentioned in my previous slide, and we've embraced the values of openness and transparency. We expect all the members of our teams to communicate with each other in an online forum, to share new results, to not be secret about that, and to share their procedures, their protocols, their reagents with each other freely and openly. And finally, to publish their work in journals that embrace the principles of open access so that not only the network, but everyone around in the academic community, in the biotech community, and in the private community can see what we have been doing with the funds that we have been provided. Finally, let me tell you about where we are. We have now identified, after 2 cycles of funding and 3 years of effort, 35 teams, including 163 laboratories, individual laboratories. We have given almost $300 million to these teams and an additional $200 million to other programs that affiliate with us, to a sum of almost $500 million, and likely much more to come in the years ahead. We have a quite diverse set of team members, quite a number of whom are led by women or early career investigators. There are 14 different countries engaged in our network, 80 different institutions, academic or research centers, and many of them have engaged in collaborations leading to new developments. So with that, I'll conclude and summarize. The problem of Parkinson's has been with us for now 200 years since its first diagnosis. There are treatments that remove some of the symptoms of the disease or control some of the symptoms, drugs that patients can take, or surgical procedures that help. But the overall progression of the disease, which is inevitably fatal, has not been changed at all in spite of intense work. And it is our conviction that by organizing an international consortium of investigators in a free and open exchange of ideas and results, we will speed the pace at which discovery is made and lead to the identification of new targets that can then be directed by therapeutic approaches in biotechnology and in the pharmaceutical industry. So thank you for your attention, and I'd be happy to take questions if that's feasible. Thank you.
Yeah, thank you very much for a great lecture. We have one great question. In your opinion, does Neuralink technologies have a potential for solving problems of Parkinson's disease or of humanity, or without using more advanced and complex approaches with using achievements of project you working on, it's barely possible?
Yeah, could you repeat the first few words of your question?
Yeah, of course. In your opinion, does Neuralink technologies have potential for solving problems of Parkinson disease?
What is that word? Neverlink. I'm
Neuralink,
missing.
the Elon Musk project. Neuralink.
Oh yeah, yeah. A Neuralink. I see. Okay, Neuralink. Yeah. I'm not aware of the details of Musk's proposal. I think with a complicated problem like this, one must use many different techniques. And the range of techniques displayed by the investigators in our network is enormous. We go from biophysics and studies on single particles or single molecules through visualization using the electron microscope or fluorescence microscopy, to genetics, to behavioral studies, to brain imaging. So we want to be completely open in the range of techniques that we will apply, in the hope that some will provide new clues that can be turned into therapeutic approaches. So we welcome, of course, other approaches, but we think the key to success in our case is by having this massive collaborative effort where we engage people in active discussions around the world.
All right, thank you. And the next question is, does Parkinson's disease of varying severity differ in genetic pattern?
That's a very good question. Some genetic forms of the disease progress very slowly and don't appear until the patient is relatively older, like at age 70. Some genetic forms of the disease appear very early. They express themselves in patients who are as young as in their 20s, and they can progress very rapidly. So there's no one category of genetic form of the disease that is either early onset, late onset, or different rates at which they progress.
All right. Thank you very much for this. We have one more question. Wait 1 second, please. Just 1 second. Is most disease are caused by genetic mutations?
Not necessarily. Some forms of cancer may not be the result of mutation. Many of them are. Of course, other forms of disease are the result of an overactive or an underactive immune system. But not all diseases have a purely genetic origin. I mean, for instance, some forms of cancer are initiated by environmental toxins like smoking. They may progress by the accumulation of mutations, or people may be susceptible because of particular mutations. But in these instances, the disease is initiated by some environmental insult.
Okay, let's go to a little bit more common, not to the topic questions. Tell us, please, about your experience in Nobel Prize award and how it was, a little bit about your experience.
Sure. Well, so my work over many years has been on the mechanism of protein secretion, which I touched on just a bit. Some of the genes that we discovered many years ago are now known to be the site of mutation in Parkinson's disease and in other diseases. But my work, until this role that I play in this organization called Aligning Science Across Parkinson's, my work has been entirely basic science. I never actually had any particular application for the research that I wanted to do. I was interested in simply figuring out how cells work in a certain process and understanding that at a molecular level. So I was happy to do that, but it turns out that a lot of discoveries that are start off as basic science can inevitably lead to direct practical applications. And so my work started with an examination of the process of secretion in baker's yeast, simple yeast cells. And we discovered the genes that are required for the traffic of proteins that a yeast cell needs to get these molecules secreted. And the genes that we discovered turned out subsequently to be seen in the human genome also. So this is a process that is fundamentally conserved over 2 billion years of evolution. And as a result of that evolutionary conservation, it became evident when the biotechnology industry was growing up that one might use yeast cells as a platform for the production and manufacture of clinically important proteins like insulin. And so I participated with a local company, and they were able to engineer human insulin, recombinant human insulin, in yeast cells. And now 1/3 of the world's supply of human recombinant insulin that diabetic patients take is manufactured in yeast. So that connection between basic science and this obviously clinically important development gathered a lot of attention for my work. And over the years, my work started to be recognized with various biomedical prizes, culminating in 2013 in the Nobel Prize that I shared with 2 other individuals who work on the same process. So that was a very exciting development. It changed my life considerably. It has given me a platform to express my views on other areas of science and the application of science. And it's given me an opportunity to speak to my friends in Ukraine today. So I'm grateful for those opportunities.
Right, thank you very much. It was a great pleasure to hear you today. I hope that we will meet in peaceful Kiev very soon. And I hope that you and me will meet and give the lecture in person.
Yes. Well, let me add that a branch of my family on my mother's side originates in what is now Moldova, was Bessarabia. And family members also were from Ukraine and from Russia, from Western Russia. So I have long planned to visit Kishinev, Moldova, but that has been interrupted by the pandemic and now this terrible war. And when Ukraine is ultimately victorious and your country is restored to peace, I very much look forward to visiting you in Kyiv or in other cities, as well as my visit to Moldova. So stay tuned.
Thank you. Yeah, we have one more question. Can we go back?
Of course.
Okay. Have you tried turning off one of the genes in the cascade responsible for Parkinson's disease?
Turning off or turning up?
Turning off one of the genes.
Increasing its level, yeah. Well, that would be complicated for these genes because, for instance, with the LRRK2 protein, the consequence of turning it up may actually result in worsening of the disease. Remember, I told you the mutation that causes the disease actually turns up the enzyme activity. So you don't want to make more of that. So you need to know more about these genes before you start manipulating the level of their expression in a human being. There are, however, examples where turning on genes may actually help in genetic and possibly in the more common sporadic forms of the disease. So this is an area that is certainly part of the work of the network. And so what we would need to do is to find those genes whose turning up or turning down can help with pathology, and then find chemicals that would cause them to be turned on or turned off, and to do so without being toxic. So we're looking for those genes whose manipulation could help. And then once we find something that we think would have benefit, we will then allow these investigators to take their discoveries and develop intellectual property, patent the discoveries, and then turn to a more commercial development.
All right, that was the last question. Thank you very much.
Okay.